We're going to discuss denaturing and renaturing DNA and the applications that it can have in research. So, denaturing of DNA is essentially just melting of DNA. So, if we look at denaturing, we'll just draw it right here. Melting. So, DNA strands melt at different temperatures depending on the bonds or the base pairs that they have in their sequences. So, if you have, let's say, just a small double-stranded molecule that has 20 GC bonds and 5 AT bonds versus a molecule that has 20 GC, or hold on a second, let's just do the complete opposite, 5 GC and 20 AT. Now it looks like since these both have the exact 25 total base pairs, they would have the same melting temperature. But they don't, because the GC bonds have 3 hydrogen bonds connecting them. So that means more bonds need to be broken in order to melt or separate, denature the DNA. So this one would have a higher melting temperature than this one. If we look at a graph that would kind of draw this, it would look something like that. So if we had the melting temperature here, which is just Tm, and then the percentage of GC bonds, it would probably look something like this. Kind of a logarithmic growth. So denaturing of DNA can be reversed, and that is through renaturing. So if we draw renaturing, just cut down here, it is basically the you anneal the, it's called re-annealing, so we'll actually write that out, re-annealing, and it is essentially just bringing back, so you lower the temperature, which allows the bonds to form between the base pairs once again, and you would have a new double-stranded molecule or the same double-stranded molecule. But an application that this could be used in research would be hybridization. So hybridization is when two single-stranded molecules of DNA from different sources, so different organisms, let's say. So if we have a human right here, and we put his DNA into a flask or a petri dish, and then we take this bacterial DNA right here, and we put it in the petri dish, and then we denature them, so they both end up being single-stranded molecules. So we have these two single-stranded molecules. You can re-anneal these, so these come together, and now you have a hybrid. So you have a hybrid between the human and the bacteria. And you can actually see the two strands, like even if these strands are completely different, they don't need to be completely complementary for these to anneal to each other. So even if there are a few base pairs that are the same, they will anneal, and then you can actually compare them as long as they have enough bases just to hold the two strands together, and you can compare the differences of the base pairs. So the extent of the hybridization can be used to measure the similarity of the nucleic acids from two different sources, and then you can assess their evolutionary relationship. So you can see how far back kind of this bacteria versus this human kind of changed their DNA, because humans used to be just a little bacterial cell like this. And then we evolved into this complex organism, whereas bacteria kind of stayed like this. So where did the DNA change that gave us the ability to grow bigger and be a multicellular organism and all of the different complexities that humans possess? And this is a great thing to use for if you were looking at a human of 2017 versus a human from 10,000 BC, right? If you're looking at a Homo sapien or a Neanderthal, you can compare the DNA from this 2017 human and see how it compares to the humans way back in the day. So that is denaturing, renaturing DNA. It's a common process that's used in genetics and cell biology labs and evolutionary labs today.